I'm delighted to introduce Frances Lefcort. Many of you know her. She's a professor of neuroscience, biology, and a number of other things at Montana State University. Frances has been working on FD for a long time. She has a personal connection with the disease. Maybe she'll tell us something about that. I can tell you that she spends some time at the center in New York with us, and those weeks or like month and a half was really fantastic. Every day we were able to discuss something else. She participated in all the clinical activities, gave us tons of ideas, and I hope she continues as excited as she has been and bringing a lot of new treatments for FD. Frances, is she around? Here.
Thank you. Okay. Thank you. Well, thank you, Horacio. Thank you, Lucy, Faye, Lainey, the board, everybody for inviting me here, and for all that you do, and for teaching me so much. Okay, so I'm here to talk about, it's really just reiterating the same message that everybody else has been saying, from what Sue said, from Horacio, Mikel, that we really all need to work together. And there's, different arms of the team here, and we represent the mouse FD team, but we really need each other because we stand on your shoulders, and it's from your insights that we do our work. Oh, and I also have to make a special mention here. This beautiful image is also the star of the show. So this is a neuron. This is the single nerve cell that's the building block of your nervous system. And of course, this is not just any neuron here. This is a gorgeous drawing from Ramón y Cajal, Santiago Ramón y Cajal, who was a famous Spanish neuroanatomist at the turn of the last century and got the Nobel Prize. And this is an image from a book that Faye Ginsburg gave me when I was here last fall. And it's full of images and of his, and it's stupendous. Okay, so where are we? We need to work together. And where are we in research? So our work as basic scientists depends largely on the work that's happening at the Dysautonomia Center. So from the basic insights in the clinical issues patients deal with, that's what we learn to focus on as basic scientists. So the goal is to then take that clinical information, translate it into model systems that we can then manipulate and try to develop treatments for the disease. So the take home really of my talk is that, again, reiterating what Sue said, is given how fast this field is moving, it really behooves all of us, FD families, clinicians, clinical scientists, and the basic scientists to be working together, because there really is great potential to translate a lot of the basic research findings that are accruing into new potential treatments. Okay, so here's a mouse, and here's a mouse's brain in living color, thinking about what we can do in mice. So our goal as basic scientists is to discover why neurons are impaired. So neurons, again, are the basic building block of your nervous system. They're cells, and we need to understand why they are reduced in number and or not working perfectly in the FD nervous system. And that's the goal of my lab. And the other major goal of our lab is then to use that information to discover therapeutic strategies for improving the health of neurons, and in the case where things are progressively dying, as in the retina, to try to prevent that progressive death. Okay, so why mice? So other than monkeys, mice are the next best model for approximating a human disease. They have all the same neurons, the same nerve cells that are affected in FD, and they express all the same genes and proteins that we do, and they work the same in mice as they do in us. And lastly, we can also make mice that exhibit many of the characteristic features of FD, but now we have a model system that we can test these novel compounds in, or novel genetic approaches like Horacio described, in a mouse first, and to learn from those tests whether it would be worth proceeding to try to test some of these things in patients. Okay, so our lab is doing a lot of work, and we have a lot of collaborations, and we have a lot of irons in the fire, but I thought I would just focus on two stories today. Quickly, they're short stories, and those are what is causing the gastrointestinal distress in FD, and if we can understand what's going wrong in the gastrointestinal system, can we develop new treatments to help? And the second story I'll tell briefly is what is causing the progressive visual impairment in FD, and can we develop new treatments to stop that? Okay, so the GI, the gastrointestinal problems, again, and I should mention both of these stories couldn't happen without the collaboration of FD families and the Dysautonomia Center and the foundation and the scientists. So this story, the gastrointestinal problems, is a collaboration between the Dysautonomia Center here at NYU, scientists in Montana State University, this is where I live, and you, all of you, FD patients. So, as many of you might know, an emerging big theme in the last few years has been the fact that the gut, your GI tract, and the brain communicate extensively. So there are chemical signals going back and forth between your gut and your brain, and they're also being affected largely by the fact you have trillions of bacteria living in your gut. And these bacteria are really, really important, okay? You would not have a functional digestive system were it not for all these microbes, these bacteria. And it turns out the bacteria are even secreting chemicals that affect our neurons, okay? So not only do you have 500 million neurons in your gut, but the gut is also innervated by other axons coming into the gut, like from the autonomic nervous system. But all these neurons are communicating with these bacteria via chemical signals, and then together they're affecting the activity of the cells that line your gut, the epithelial cells that line your gut, and they're all communicating with the brain, and it also can involve the immune system, okay? So if there is some kind of breakdown in this communication, for example, in FD, we know there are fewer neurons in the gut. There are fewer neurons in general in the autonomic nervous system and the sensory nervous system. We also know many FD patients take a lot of antibiotics, and those antibiotics are going to affect the makeup, the composition of these bacteria that live in your gut. So is there some kind of imbalance in this gut-brain axis, and can we figure out what it is, and can we intervene to improve the function of the GI tract? So this is a collaboration, as I said, with people at MSU, Montana State University, and the guy leading the effort here is Seth Walk. He's a microbial ecologist, and this is really his forte, is understanding how the gut microbiome works. But here's your part. So we started with you, and we got stool samples from you, collected at the Dysautonomia Center, and then Seth and his lab analyzed them, and sure enough, there are, and this is a hard graph to understand, but just look here, these are, we got stool samples from FD patients and a healthy relative, and you and your healthy relatives have significantly different microbial composition. So the microbes in your guts are more similar to each other than they are to your non-FD relatives, okay? So that's already an important clue that there's something different in the microbial composition, and if we can figure that out, which microbes are different, perhaps we can intervene. So we also collected stool samples from our mice that have FD, and they too were significantly different from their litter mates that did not have the familial dysautonomia mutation. Okay, so that's good news. We are recapitulating this in our mice. So the take-home of this story is going to be that since the microorganisms that live in the intestine help harvest energy from food, we are hypothesizing that alterations in the composition of this microbiome, or which microbes are in your gut, are exacerbating some of the metabolic problems that can occur in FD and creating some of the GI distress. And we're hoping that if we can figure out which microbes are different, perhaps like taking a probiotic, we could say, oh, take microbe X, Y, and Z, and that would restore a more normal gut flora and minimize some of the gastrointestinal problems. And if any of you are interested in this, I'd be happy to talk more, because we've really been studying the GI tract in our mice intensively, and there are a lot of interesting differences. Okay, and then the last story is just what is causing the progressive visual impairment in FD, and can we develop new treatments to stop it? And here again, this story, we could never have worked on this were it not for your involvement and of course that of the Dysautonomia Center. So the first clue for this work really came from Dr. Carlos Mendoza Santi Esteban, who is an ophthalmologist, and of course, as Dr. Kaufman alluded to, has been showing this progressive decrease in a specific cell type in the retina, the retinal ganglion cells. And what was really interesting is that the pattern of cell loss, it wasn't universal or wasn't uniform across the retina. There was one part of the retina that was more impaired than the other part of the retina. And the part of the retina that was more impaired is the part of the retina that looks at the central part of your visual field. So that's where you pay most attention to. And so that part of the retina requires more energy than the part that is looking at the more peripheral parts of your visual field. So that, when you see that difference in survival, depending on if you're looking at if you're part of the more metabolically active retina versus the metabolically more quiet retina, that's a big red flag, okay? And in fact, it turns out there are other optic neuropathies, so visual impairments that are due to mutations in genes in a region of the cell called the mitochondria. And the mitochondria is the energy source of the cell. So if something's wrong in the mitochondria, you don't have enough energy and cells can die. So when we heard this pattern of retinal cell loss, we were thinking, wow, mitochondria. Okay, so here is a little tour of the cell. And here's a cell, here's the nucleus where the DNA is, but out here in the cytoplasm of the cell, you have these mitochondria. And these are, you know, the LeBron James of the cell, okay? I mean, if you don't have LeBron James, you know, you're not going anywhere. If you don't have mitochondria, you don't have any energy. You're not going anywhere, okay? So this is a really, really, really important region of structure within the cell. Okay, well, so here's the second clue, which came from an FD family. So it was at one of the FD research conferences, and one of the parents came up to me and said, you know, and I think I had mentioned mitochondria in my talk, and he said, our son, before we knew he had FD, his pediatrician thought he might have a mitochondrial problem based on some of his symptoms. So he was sent to Emory in Atlanta, where they have a mitochondrial genetic disease lab, and they measured his mitochondrial activity. So these, you don't have to worry about these numbers, but these are different structures within the mitochondria. And he sent me his report from Emory, And it showed, sure enough, that these really important proteins that function in the mitochondria to make energy were significantly reduced in number, okay? So that was like, whoa, red flag. So these mitochondria, we seem to be impaired, and so we have spent a lot of time trying to understand if that's true in our mouse model, and if so, can we improve mitochondrial function? And so in particular, we've asked this question both in the peripheral neurons that mediate pain and temperature sensation, and those sensations are reduced in FD patients. And we're really interested if that is causing the visual impairment, part of the problem causing the visual impairment. So here too is another beautiful drawing from Ramoni Cajal. And there are several cell layers within the retina from the book that Faye gave me. And there are several really interesting cell types within the retina, but the cell we care about that seems to be most vulnerable in FD are these retinal ganglion cells, and Dr. Kaufman mentioned them as well, because these cells send their connections out of the retina to the brain. So this is the output cell from the retina, and so if visual information is going to get to your brain, it's got to go through these cells. Okay, so we have made two mouse models now for FD where, and we've studied the retinal phenotype. Like, do we get retinal degeneration in our FD mice? And the answer is yes, and one study we've published, and another study is in revision right now. But the cool thing is, I took the slide out because I thought I had too much data, but I'll just mention briefly, when I saw Dr. Kaufman's slide of the degeneration in the ganglion cell fiber layer, our curve mimics it absolutely identically. It's the steady decline early and then a slower rate of degeneration. So, I mean, we really have a perfect model, and now we start to get the degeneration 1 month after the mouse is born. And so we really have a great model where we can test therapeutics in now. Okay, so can we improve mitochondrial function? So as Sue mentioned and Dr. Kaufman mentioned, yes, so we are very hopeful. So this was, again, a chance interaction with Mike Brownstein at one of the FD meetings, and again, I think he had heard me say that we were thinking there's a mitochondrial problem, we think our cells are under stress. He said, gee, there's this drug, BGP-15, why don't you test it out? It's supposed to help in situations where cells are under stress. So we did an experiment where we tested his drug, and we tested it both in a culture dish, and it worked really well in a culture dish. And so we thought, wow, let's see if we can inject it into a mouse and see if we can prevent this specific cell population, the pain and temperature sensory afferents, from dying. And we did. So here is just, I'll show you 3 quick images. So in red, so these are normal neurons here from our mice. These are neurons that lack the protein ICAP. And so what you can see in red here is a marker for mitochondria and healthy mitochondria. So here in the control cell, you see lots of healthy mitochondria. Here in the mutant neuron, we don't have anywhere as many healthy mitochondria. But here in neurons that are exposed to BGP-15, this drug, we've got healthy mitochondria again, okay? And these were neurons that grew up without ICAP protein. This is another measure of health of neurons, and for the scientists in the audience, this is measuring reactive oxygen species. So if you have elevated reactive oxygen species, it's an indication that cells are under stress and their mitochondria aren't working well. So the BGP-15 corrected, reduced the levels of the reactive oxygen species, so this was really exciting. And then this was the last experiment where we looked at cell numbers. So normally we have fewer of these pain and temperature afferent neurons in our mouse model, and we could restore to almost normal numbers just after 5 days of treatment with BGP-15 in the mouse. So we are hopeful that either this drug or some variant of this could be tested eventually in FD patients. And we're now doing studies to try to see if this will work in adult mice on the baroreceptors, the sensory neurons that regulate blood pressure stability. Okay, the last approach I'm just going to mention very quickly is another approach we're going to try to develop in our lab, is to try to do what's called gene therapy, to reintroduce a normal copy of the ICAP gene into adult cells. And the cells we want to do this in, the first cell population is the retina. And this is how far we've gotten. We found a virus, an adeno-associated virus, that will infect, and these are shown here, the retinal ganglion cells. So we can get really good, this virus is the best one we've tested. These little green cells here, shown here also in red, are the retinal ganglion cells, the cells that die in FD. And so we found a virus that will infect these cells, and you can see here other views, how many cells they can affect in the mouse retina. And now the goal is to insert the ICAP gene into this virus, and then we're going to inject that into mice of varying ages to see if we can prevent that progressive death of the retinal ganglion cells. So to summarize, as Sue sort of stressed, I really think this is the time to be all talking to each other and to working together to test combinations of some of these approaches. So Dr. Slagenhaupt has got a great strategy for correcting the mutation with this and improving splicing to increase levels of ICAP protein. That really should be tested. We are really interested in testing BGP-15 or other compounds that could improve mitochondrial function. And then this last approach of gene therapy, perhaps trying to introduce, using viruses, a normal copy of the gene. And I should mention that this gene therapy is now working really well in another retinal genetic disease, retinitis pigmentosa. So there are really reasons to be hopeful, actually, about all three of these methods, and especially because we can combine them. And as Dr. Kaufman alluded to, it could be in different tissues at different stages, one of these treatments is better than the other. And so that's a really interesting tack to take, because it does seem that different neuronal populations are more vulnerable to dying at different stages, whether development or in the adult state. Lastly, I just want to acknowledge the inspiration for what I do are two first cousins of mine from Montreal. My family's originally from Montreal. Barbara and Michael Cronick, who I grew up with and struggled with FD every day of their life. And I want to thank everyone. I want to thank all the great people in my lab at MSU. I want to thank great colleagues at the Dysautonomia Center who hosted me last fall. I want to thank the foundation who has funded me in the past, and the NIH, your tax dollars at work that have really supported a lot of our work. So thank you very much.